This is part 2 of Chapter 7. We're continuing our discussion of viral diagnostics. We just talked about several cell-based assays, immunofluorescence, immunohistochemistry, plaque assays, and some of the other tests I wanted to talk about are serum-based. So these would include things like the ELISA test, Western blot analysis, agglutination reactions, lateral flow immunoassays, which are kind of pregnancy test style assays. And then also want to talk about some of the molecular biology based tests where we're looking for viral nucleic acids. So let's talk about some of these serum-based assays, and this falls under that category of serology. So remember, we had these three general approaches to diagnosis: tissue culture, which we already talked about; serology here, which is any test utilizing blood serum, liquid portion of the blood, and then the molecular type tests. And so these serum-based tests are often looking for the presence of antibodies in an infected person. And just to remind you, we have different types of antibodies that your immune system produces during an infection: IgM, which indicate an early infection, versus IgG, which gets produced later in an infection. So, which type you're detecting or looking for might give you some indication of either, how far along a person is in their infection, or you might need to use a slightly different test depending on where you think they are in their infection and what type of antibody you have available to test for. All right, so let's start with the ELISA test. It's a very common antibody-based assay, and there's two different variations on this I'll show you, but as long as you get the basic gist, should be fine. So there's what's called the sandwich ELISA and an indirect ELISA. And I'll start with the sandwich ELISA, and I think you'll see why it's so named. But these type of tests are being carried out in 96 well plates. Let me just jump ahead a couple slides to show you what that looks like. So a little dish like this where you have these tiny little wells, right? So that's what you're seeing here. This would be individual wells on that plate. And so in the sandwich ELISA, we've coated the bottom of the wells, the plastic at the bottom of those dishes, with antibodies that have been designed to recognize and bind to a specific viral antigen. So you would take your serum from your patient or your individual being tested, and you would add those to the wells in the plate. And the idea is if you have an uninfected sample, there's no antigen to bind to those antibodies. If you have an infected sample, there is antigen that binds to those antibodies. Right, so you add your sample, allow for binding to occur, wash everything out. If the antigen sticks, it won't get washed out. Then we'll add a secondary antibody that has also been designed to recognize that same antigen, but now that secondary antibody is linked to an enzyme or a fluorophore, so you can actually see it. And now, if there was viral antigen present, you're going to get a color change or a fluorescence in that particular well. Okay. And so basically, that name sandwich ELISA is coming from the fact that you're sandwiching the viral antigen between two layers of antibody in this case. In an indirect ELISA, it's sort of the opposite approach, in that instead of coating the bottom of the well with antibody, you coat the bottom of the well with viral antigen. And now in this case, you're going to try to fish out any antibody from infected serum. So, if an individual has been infected, they're going to produce antibodies, or their immune system is, against that virus. So here in an uninfected sample, right, that individual has not produced any antibodies to identify that antigen, so there's no antibodies that stick. Whereas in the infected sample, there are antibodies that will bind to that antigen. And then again, we need a secondary antibody that's going to allow us to visualize what's going on In this case, the secondary antibody is an antibody that's designed to bind to the primary antibody, right, the IgG or the IgM. And so if we had our infected sample, that secondary labeled antibody will stick, and we'll get a color change in that well. If, in the uninfected sample, we won't. And so you'd end up with something like this in your plate, right, where color change tells you, yes, that was a positive sample, no color change, no infection, negative sample. And sometimes it can be somewhat quantitative as well, right? So the darker the color may mean you had more antigen or more antibody present in your serum sample. This is what the machine looks like that would then read the plate, and obviously you can see it with the naked eye, but will actually quantify the intensity of the color change. So again, this is a very common test, common equipment. We have capabilities to do this here at Pitt State. So that's the ELISA-based test. Another common one that's kind of an older test is the Western blot, but it does still get used today. And so Western blots are basically tests based off of running proteins through a polyacrylamide gel. So you can separate proteins on a gel based on their size, just like you would DNA samples in an agarose gel. So the setup is a little different. It's usually a vertical gel instead of a horizontal. But you load your protein samples in these wells at the top, and as they migrate through the polyacrylamide, the smaller proteins will move more quickly and the larger ones more slowly, and we get that separation based on size. And so because it's difficult to really do any kind of assays in that polyacrylamide gel itself, in a Western blot, you then transfer, after you've run the gel, you transfer the proteins from the polyacrylamide onto a membrane. So you basically lay a membrane on top of the gel and apply a current in such a way that you move the protein bands out of the gel onto the membrane. So you now have this essentially membrane copy of your gel, and that membrane is easier to deal with. You could add, in the case of these Western blots, you can add antibodies to specific viral proteins. If that protein was present in the gel, the antibody will stick to that specific band on the membrane. And using similar techniques to what we've talked about in previous assays, you can get a color change or a fluorescence that lets you see, which bands on this gel, which proteins might be viral proteins, or which samples might be infected versus uninfected. So again, it's an older technique, but it does still get used occasionally today. The next one I want to mention are agglutination reactions. These are pretty common. These take advantage of latex beads that have been coated with viral antigen, and this is basically another method for trying to fish out antiviral antibodies out of a patient's serum, sort of like the indirect ELISA that we talked about. So the idea is you would mix these beads with your serum sample. If that sample came from an individual that was infected with the virus, they would have antibodies that would bind to and recognize that antigen. And because the antibodies are branched and they have these two arms, each of which can bind antigen, you get these clumps of these beads that you can actually visualize on a microscope. So if you get this clumping, that means that you have a positive result, right? That particular sample contained antibodies against the virus, meaning they had been or currently are infected with that virus. The last of these serology typed assays is the lateral flow immunoassay, and as I mentioned, this is similar to a pregnancy test. In fact, this is basically exactly how one of the pregnancy tests, dipstick tests would work. And so the idea is you have this membrane here with these specific zones, and you would add your patient sample, your serum, to one end of this membrane, and as it gets wicked through, all your molecules in that serum are going to pass through these different zones. So if you think of this in terms of the dipstick, right, you've got your membrane here where you place the sample, everything's moving towards the bottom here, and you'll have a sort of an indicator area on this test. I'll show you how that works. So next to the sample well, you have what's called the reaction zone, and here you'll have some colored beads, blue beads in this instance, that are coated with antibodies that have been designed to bind to a specific viral antigen. So if that serum sample was positive, right, viral antigen is going to stick to some or all of those beads in the reaction zone. Those beads then get pushed along into the test zone just by the wicking action of the fluid in the serum, carries it into the test zone. The test zone has antibodies anchored that will also recognize that same viral antigen. So now if any of those beads picked up antigen in the reaction zone, those beads will get collected by the antibodies here in the test zone. If the serum sample was negative, right, none of those beads would have picked up antigen, and they won't get stopped in the test zone, and they'll just keep flowing on through. But if they do get collected here, then we get a line or a spot, right, in the test zone. And then you'll also have what's called the control zone here, which will contain antibodies that will just collect any of the empty beads that did not get picked up in the test zone. So that would just show you that the test actually worked, right? That material flowed all the way through. So in this case, if you just get one spot or one line here, that would be a negative result. Whereas if you get the two lines or two spots, that would give you a positive result. So the big advantage to this is it's kind of idiot proof, right? You just need your serum sample, just put the drop on there, wait a few minutes and just look and see, do you have one spot or two, one line or two? All right, so to finish this out, let's talk about just a few different assays that are really considered molecular biology type tests. And again, these are all looking for viral nucleic acid specifically. So the most common of these would be PCR, polymerase chain reaction. I know if you've taken genetics, you've learned about this, so I won't go into all the details of PCR again. But basically, PCR is this primer-based assay, right? So to carry out PCR, you have to know something about the sequence of the viral genome itself, because you have to be able to design these complementary primers that would bind to specific virus sequences. If that virus sequence is present and the primers bind via PCR, you can basically make billions of copies of that viral sequence. So it's like a copy machine, right? So you put in, maybe some very small amount of viral DNA, and you get out billions of copies, so making it very easy to see. So again, if your serum sample contained viral DNA, this amplification process via PCR would work. If it doesn't, then it won't. And so PCR is a DNA-based test, so what about viruses that have an RNA genome? Could you still use PCR in that case? And yeah, you can. There's a technique called reverse transcriptase PCR, RT-PCR, where you would take your viral RNA, do a reverse transcription in the test tube, basically to make deep DNA copies, and then you could do your standard PCR on those DNA copies. So that can be done as well. And one cool thing you can do with PCR is you could actually design it so that you could test for lots of viruses at the same time. And this would be considered multiplex PCR because you would have to use multiple sets of primers, right? So you'd have one set of primers designed to recognize one type of viral DNA or one virus. you'd have another set of primers designed to recognize another virus, and so on. And so you just add all these primer sets to one sample and see what gets amplified and what doesn't. And then, how do you actually get a result? Well, if you've done any PCR before, you basically have to run an agarose DNA gel to see the products of the PCR reaction. And there are some newer, sort of fancier types of PCR. For example, there's what's called real-time PCR, which is a quantitative PCR. So I'm not going to go into all the details here, but in traditional PCR, it's basically just a yes/no test, right? So did a particular primer set amplify something? If yes, then that meant that virus was present. If no, then maybe the virus wasn't present or the PCR failed for some reason. But in real-time PCR, it could actually quantify how much of the viral DNA was present. So it wouldn't just be a yes/no, it would be a yes, and the sample was very mildly infected or very strongly infected. There was a ton of viral DNA there. So you get these curves that basically allow you to, as I said, to quantify the amount of initial products. These are called amplification products, or amplification plots. Anyway, I don't want to get hung up on the details here, but just be aware there is quantitative real-time PCR that is pretty commonly used these days. Another sort of variation on this type of nucleic acid testing is what are called DNA microarrays, or sometimes they're called chip-based assays. Gene chips, sometimes you'll see them called. But this is essentially, it's like a little microscope slide sized piece of glass, and you will have short DNA sequences spotted in these teeny tiny microscopic spots on that glass or sometimes silicone chip. So each one of these little spots here would correspond to a different DNA sequence. These short DNA sequences are called oligonucleotides, or just oligos for short. And so, each one of those spots could be DNA from a different virus, for example. And so the idea is you would take your serum sample, isolate the DNA, you would do some PCR, maybe multiplex PCR, to try to amplify as many different potential virus sequences as you could. In the process of doing that PCR, you can incorporate some fluorescent labels into any DNA product that gets made, and then you would introduce that to your microarray, to your chip. And basically any place that there's complementation, complementary nucleotides between the DNA that you amplified by PCR and the little oligo on the chip, your DNA will stick, right? And so because those chips are designed, researchers know, okay, well, yeah, this oligo corresponds to influenza virus, and this oligo corresponds to COVID-19, or whatever the case might be. So you could say very clearly, yep, this individual is infected with virus X, Y, and Z. So those are pretty cool, and there's all kinds of other very useful applications for microarrays, research applications as well. And of course, another approach would be just to do DNA sequencing, right? Nucleic acid sequencing. And we have some very fast, what are called high throughput sequencing methods now, where you could sequence an entire viral genome in a matter of hours in that case. In some cases, depending on the virus and the size of its genome. So as DNA sequencing has gotten cheaper and cheaper over the years, this is really a more viable approach for identifying viruses. And the nice thing, if you have the whole genome sequence, is, not only are you infected with this particular virus, but you have this subtype, right? Or your infection contains this unique mutation, something like that, that could be very useful information to have. And any type of sequencing-based approaches always rely on this subdiscipline of biology called bioinformatics, which is basically using computers and computer science to try to organize and interpret sequence data, right? Because you end up with lots of data, right? Tons of A's, G's, T's, and C's. So trying to make sense of all that can be a challenge. All right, so I know there was a lot of information. Thanks for sticking with me.